Those of you who've been involved with GenRe-Mekong will know that besides collecting blood spots, we also ask people to give us some details on the travel history and you know, where more precisely they live, in order for us to be able to do additional epidemiological analysis. So Professor Richard Maude is the head of epidemiology here at Maude, and his team has been working on some of these epidemiology data sets that were collected alongside the blood spots for GenRe-Mekong. And so he's going to tell us a bit about the work. Richard, you're...
Thanks, Oliver. Yeah, so I've watched this wonderful project evolve since the beginning, back in 2014 with the inception. I'm feeling old now. So yeah, I've been asked to talk about this title. And what this is essentially is building in other things into the project, and then also bringing other information in to get added value from the data that's collected. So I'm going to talk a bit about mapping malaria, and then about this case record form that we've built into GenRe-Mekong, and then just some examples from 3 countries. I'll start with this. So we did a systematic review a couple of years ago of all of the different research projects around the world which had collected CALCH 13 marker data. So then we've mapped those projects for the time that the samples were collected. And you can see that over time, the number of different projects collecting samples goes up, and the geographic extent of the data increases. Just repeat that. But there are lots of gaps in the map where there's no data. So lots of places that don't have any genetic marker data. And that's because this was dependent on primarily research studies where there was a study site for a few years, which came and then went. So there isn't a clear, consistent picture over time for a lot of the world. You can see the GMS is a bit better because there's been so much focus over the years in the GMS. So the data becomes quite dense in recent years. And what GenRe-Mekong has done wonderfully is transition from partnering with research projects to now becoming part of routine activities, so part of surveillance. So that's able to fill in these gaps in the map and continue to do it over a long period of time, which is quite a special thing. One of the other challenges that we highlighted in this particular review is the data from those research projects was published on average 3.5 years after the result was available. So after the data was collected. So the sample collected, and then 3.5 years till the information was made public. So hopefully 3 to 6 months is much better than that, and that's the way things are going. So for mapping malaria, what's traditionally done is, and what we've seen here is study sites by health facility. So if you imagine this as a district in a country, what we try to do at GenRe-Mekong is build in collection of where the patients live. So these dots on the second box are villages, so we're mapping at least their residents. We're also interested to know where they've traveled, because often these people have been somewhere and caught malaria in a different location. So we're trying to capture where that place is. So in recent years in the GMS, that's been in the forest, which is why I've got these dots in the forest here. Some villages still have transmission, but that's changing. And then over on the right, we can then start to also map out how we think that the parasites are spreading geographically. Depending on the level of detail captured and how good this information is, you can go quite high resolution with this information. So we're going from just the health facility to the address, capturing some travel information, and then both with the travel patterns and the population genetics combined, we can start to map out this spread. This is the situation in a lot of countries. I think these pictures are mostly from Cambodia. These are people in the forest who are spending different periods of time, up to a couple of months in some cases. And they're staying in places like this hut where they're very badly protected from malaria, and there are clusters of transmission in the forest. Sometimes these are quite far away from where they live, and they're certainly very far from the hospital or the health facility where they're diagnosed. So we piloted this short 2-page questionnaire, initially in Cambodia with Dr. Sib and his team. We did quite a lot of work in the villages to work out the best way to ask these questions. And we've included quite simple information on age, gender, occupation, where they work, because in some cases that is where they're infected, their address in some detail. So we've standardized the way that the geographic information is collected. A lot more information about travel, including specifically to the forest, but also other types of travel, and trying to map as well as we can where they're traveling to, including to other countries. So a few years ago, we had a small grant from the Wellcome Trust where we developed a platform where we did a simple analysis of this data and we operationalized it. So it was done routinely. There's a delay because the data has to be captured and cleaned and organized. But once that's done, we did some very basic mapping and graphing of the results and then gave access to some of the national programs. So that platform still exists, but the grant finished a few years ago. But we've got data showing all the different locations where we have both blood samples for genetic analysis, but also the case record form. And you can see that from early on, some of the partner studies, we also have Bangladesh, India, a few samples from Myanmar, as well as the GMS that we've heard about. So I think we've got at least the last count of more than 14,000 of these case record forms completed. So this is one of the early examples. This one's from Vietnam. So this project was led by NIMPE. We supported them to run what is essentially a case control study. But what NIMPE did is they took people presenting with malaria to health facilities, and then they would recruit the person with malaria, and then they would match a control by age and gender in each of the health facilities. And then we were able to compare people with malaria with people that didn't have malaria. So the advantage of that is you can identify both demographics of people with malaria compared to the general population, and also where they've traveled to. So what's different about these people? Which places are they going that people that don't get malaria have not been to? So here we've got a population pyramid where you can see a huge excess of working age males amongst the malaria population as compared to the general population, where that isn't the case. And that's reflected in a lot of the other countries around the region. We're able to map where people travel from and to just before they get infected and during the period that they're infected. So this map in the middle is just some of those results. You can zoom in and see in quite some detail where they've been during that period. By combining data, the figures here on the right, the communes that are colored in are where they live, and these pie charts are where they're traveling to. So we can see people traveling to the forest, so these pie charts here, and they're traveling from lots of different communes to the same part of the forest. So that's where they're probably being infected. So this was shared at the time with NIMPE. There was a lot more transmission back. This finished in 2019. But it's actionable evidence, finding where people are being infected. We had an early partner project with the Bangladesh National Programme. So this was a large study with over 2,000 cases from which we have blood spots. So these blood spots are then analysed through GenRe-Mekong. But we also included the national surveillance data from the country. So we've got, this is the recruitment. So these are the study sites. And on the right, we've got the numbers of total cases reported in the country. And you can see all these lines on the right, which are the travel routes of people during the period that they were probably infected with malaria. Combining all that together in a model, you can start to predict where we think people are being infected. So the sources of transmission in a country. So the red patches on this map in the middle are where the most likely places are people are getting infected, which is not always the same as where the highest incidence is, because a lot of times people travel to these places for a reason. They get infected and then they go back home and then they have their test, the malaria test. So we also analyzed the samples, the blood samples, and looking at the parasite genetics, we were able to split this quite heterogeneous parasite population into two groups. So this map on the left, there's two clusters of transmission. There's the north of the endemic area and the south, and they're quite separate. The parasites in the north are quite similar to those in India. And the border with India is very porous. People travel across it all the time. And in the south, they're more similar to parasites from Myanmar. So they share mutations with parasites that have been sequenced in Myanmar. So fortunately, there was no evidence of artemisinin resistance. So there's no K13 mutations, and there's no genetic background at the time. This finished in 2017, so the country's very interested to repeat this. They're quite concerned that they might be starting to get signals of artemisinin resistance. The other thing you can do with this sort of data is you can, again, in a simple model, you can start to estimate how much importation there is of parasites. So the map on the right is by sub-district, and it's looking at the proportion of parasites that we think are imported in each of the areas. So the other thing we're able to do in Bangladesh is bring in population movement from mobile phone data. So people that use a mobile phone, their location is tracked in real time by the companies. That data can be aggregated and anonymized, and they're used as another measure of travel. This is a very active area of research. You can see the map on the right that I've made was coverage at the time of this project. So the dots are where you have cell phone signal. That's getting better over the years, but at the time, this part of Bangladesh, there's lots of places where there's no dots, there's no signal. You can see, so like, for example, Myanmar, there's huge gaps in coverage. Thailand was a lot better off. But there's value in combining these data together because it gives you very rich information. So here we've got travel surveys. This is the information that we get from asking people where they've been. If we include the population genetics with this heat map of importation, and then with the cell phone data as well, you can start to map out in quite some detail where parasites have come from and where they're spreading to. So this is a modeling analysis, and it uses big data. It was picked up by the press at the time, but the way that we do it is it's very ethical because this cell phone data, as I've said, it's completely anonymous and it's all aggregated, so you can't identify any people. We're not tracking individuals. So one thing that we've been trying to do is support this transition by country. So this is the WHO guideline, the monitoring, surveillance, monitoring, and evaluation reference manual, which recommends that as countries approach elimination and they have fewer cases, to have more and more geographically granular data. So a higher resolution mapping of the cases, and also to collect information about where they've been, so you can classify the cases as imported or local. So Bangladesh, as with other countries, are very keen to do this. They're on the journey. They started out like this with large areas with numbers of cases by area. This is the central database, the way they have the information organized. What we've been doing is supporting them to change that to individual level with a high quality map of all of the villages, so they can then map everything by village. This becomes particularly important as the cases drop, to find those few clusters of villages where there's still transmission. So we're trying to get them to the point on the right, which is the number of cases by village. You can calculate, dividing by population, if that's collected as well, you get API by village, which is really good for then targeting both data collection, but also interventions. So you can see the cross here, that's the health facility in the middle of that area. That was the state of their data collection original, and then we're transitioning to this really fine scale granular data. Just to finish with this project in Laos. So Simpe have been working with LOMRU to routinely collect these case record forms along with their blood samples. So we've had a PhD student who's been supporting this with some of the analysis. So this is over 6,000 people with malaria, and we've been collecting this travel information, the demographic information, and then mapping the results. So this is where people are traveling from and to within Laos by district on the left over this period. And then what we're able to do with this larger data set is look over time how stable this is. So do people continue to travel to the same places over time, or does this change? So we've got districts listed here, and then on the bottom is by quarter, and then the darker the color is, the more travel to those particular districts. So you can see it's not so stable. There's certain places where there's more travel in certain years, and it changes over time. So one important thing during this period is obviously COVID happened. So the pandemic resulted in changes of people's behavior and changes in how people travel. What we found in Laos is interestingly, the overall amount of travel of people with malaria went up. So these are in more rural areas, particularly the people going to the forest. From this data, the proportion went up during COVID. And then, so this is the stringency of the lockdown measures. You can see this line here on the left. Anyway, this line. So the lockdown was very strict at the start, then it was relaxed, then it was again strict later on. And the travel at the top just continues to rise throughout that period. So remember, this is not the general population. This is people that had confirmed malaria, which is very different. The other good thing about this information is you can see it's the same group of people that were infected over time. So if you look by year, it's still majority male, majority working age. That's not changing. And it's also the same occupation groups that get malaria. That's also not really changing. So you heard about the outbreak in Attica province. So we've also done some analysis for that. So the travel information and the demographic information that was collected, we've done an analysis after the outbreak. So you can see here that the line at the top is the total number of cases. So this is in the province. So there's a huge peak here in, within that red box. And then the colors are the different parasites. So there's a resistant strain. There's in fact 3 different strains that are part of this outbreak, and they all appear during this time, during that period, and then they go away. And within one district, because of the information we've collected, you can see that the people affected by the outbreak were almost all living in this one district. It was also the most popular place for people to visit during that particular period of time. So it was the top rated out of the whole country, the place that people travel to the most with malaria. So people that got malaria, they went to this one district more than anywhere else. What we could also see from the travel information is that they weren't traveling out of this area in large numbers. So the outbreak stayed focused in one particular district and didn't spread. So we can see that the people affected by the outbreak, it was the general population. It's similar to the overall people with malaria, the same age and gender breakdown, the same occupational risk groups. It was general spread in the population. And then if we look at what was thought to be part of the problem, if we look at rainfall over time, there was no change during that particular period in this area. So it wasn't driven by rainfall, it wasn't driven by climate. But it looks like there was an overall increase in seasonal transmission. So vivax also went up at the same time as the falciparum outbreak. There was some clonal expansion of these strains, and it remained confined in this one area because of not very much travel, which is a good thing and helpful for the national program to know. So the other part of this questionnaire is about asking people whether they actually use a mobile phone. So this is, again, the population with malaria, because we're wondering if this mobile phone data, like for Bangladesh, would be useful for this sort of analysis. And for Laos, we're finding that the proportion of people with malaria that have a phone is quite low, whereas the general population, it's a lot higher. So this sort of routinely collected mobile phone mobility information is probably not so useful for Laos. It's very important to know that before trying to use it to generalize. And we're finding that the more malaria there is, the lower the ownership there is. So parasite genetic information can be greatly enhanced by some additional simple data. So we can do high resolution mapping. It's really good for spatial targeting of responses, which can be good for making things more efficient, saving budget. Demographics tell us who was infected with which parasites, the travel, where were they infected, and why was that? And then just to note that we can also cross-validate between the travel information and the population genetics about where we think the parasites are coming from. When the parasites are clonal, sometimes the travel information is the only really granular data that we have, because the parasites all look very similar across the map. And then to note the major limitations of travel surveys, people have to trust the person asking the questions. They have to remember the information, of course, and it takes time and resources to analyze and process this to then be able to give it to the national program. Thank you.
Very much, Richard. I think we have time for at least one question. Anybody would like to volunteer?
Thank you, Professor Richard. Very interesting research. Just in terms of the ethical manner, because you follow the mobile signal and the movement of people. Actually, in Vietnam, we very interested to track the two objects. One, malaria patients. If they go to the receptive area, it will be very dangerous. Yeah, because it brings reservoir to the endemic to the free malaria area, and the second from the pupil come from non-endemic to the endemic area. So my question is, how possible if we can check that people and warranty that with the privacy?
Yeah.
Yeah, so thank you.
So this changed a lot during COVID because some countries, including Thailand, started to do that routinely. For research, it's a whole different matter. We're a lot more careful. These things always have to be approved by the regulator, the mobile phone regulator in the country, which is one of the problems. And then the company has to be willing to share the information. For a national Ministry of Health to use that precedent's already been set. It's whether they can get access to it. I think important though is the engagement with the communities because they need to understand what's happening to sign into it. And there's projects in the past where that hasn't happened and there's been a big backlash in the media against it.
I think we can take another question. Yes, Alfredo.
And thank you for the nice presentation. My question is, what is the action behind this data? I'm working in Mozambique. We are trying to do similar work in the south where there is low transmission. And when we talk with the National Malaria Control Program, it is not very clear what is the reaction to all this information. I don't know if in this region there are more clear plans and recommendations. Thank you.
Yeah, there is. So things are changing a bit as the cases come down because the countries are starting to collect this routinely as part of case classification. So it's essential to know about importation of malaria. That's one of the key questions. And then the other information for a particular scenario like the outbreak, it gives you more granular information. That's very new. That hasn't been done before. So we're still learning in this region too.
Dr. Silva is asking for.
Just one last question. You said that the most likely is not linked to the seasonal, but some people say that the relapse of virus is linked to the seasonal. So when you analyze both malaria case, will be the virus is there. So there is nothing happen.
So for this one, part of it is the seasonal peak, but there's something on top of that.
Yeah, thank you so much for presentation. Very important and useful for us because for this research, like know about the history travel right now in Laos, Kham province, Nakai district occur outbreak virus, and I saw the map, the travel from Pubong to Nakai, but we don't know this is a relapse or reinfection or not. But we conduct the DBS to send the laboratory, and next month we have the result. Yeah. But important for us because when we classify 137, we interview them and they told us travel from southern part of Laos, but not exactly. And finally, we classify malaria indigenous